Hello and welcome to our presentation about the effect the season has on wing melanization of Pieris rapeyi in North America. Pieris rapeyi are an invasive species to North America and are commonly known as cabbage white butterflies. This name comes from the appearance of the butterflies with a white ring and sexually dimorphic eye spots. Females of this species have two eye spots, the anterior M3 spot and CU2 spot, whereas males lack the CU2 spot. We hypothesized that season would have an effect on the area of wing melanization in the North American population, predicting that wing melanization would be increased in the spring season. This prediction was made due to the assumption that wing melanization is used for thermoregulation in butterflies. Brianna will now talk a little bit about how our data was collected.
For the materials and methods for our experiment, we used multiple websites and image processing program. One of the websites was idigbio.com, where we obtained all of the images of our butterflies. And another website was geolocate.org, where we verified the location of our butterflies. We used ImageJ as the image processing program, where we measured multiple aspects of the butterfly, with our experiment focusing on the apex melanization, or the tip melanization, and the M3 wing spot area on both of the wings. Altogether, we had 123 butterflies in our experiment, 66 of them being sorted into the summer category and 57 of them being sorted into the spring category. And now on to the results with Lydia.
Once all the measurements for our wings were compiled, the means of those melanized areas were compared with standard error. The M3 spots for both the left and right wings actually fell within the standard error of each other and therefore were of no difference between the samples. The apex melanization, however, did have a difference, with that mean melanization larger for both wings in the summer sample when compared to the spring. Both trends you can see in graph 1 on our poster here. The means of those samples for all melanized areas, the apex and M3 spot, were then put through a two-tailed t-test of no difference with an alpha value of 0.05. Not surprisingly, the null hypothesis of no difference did hold true for the M3 spot of both wings, with probabilities far exceeding the 5% threshold. For the apex melanization, though, we were able to reject the null hypothesis and state that there's a difference between the summer and spring samples. The t-test probabilities are laid out in table 2. So in the end, our hypothesis did hold true, but only in part. Season does appear to affect the apex melanization, but it does not have a bearing on the M3 spot. Our prediction of increased melanization for the spring was incorrect. In fact, we actually see the opposite, where the summer butterflies had an increased area of melanization versus the spring butterflies. The reason for that at this time is unknown, but it would be an interesting factor to dig into and research different aspects that could be why that apex melanization actually increases in the summer. As for the M3 spot, it's not unlikely to consider that since it is involved in sexual selection as well as predation, that those factors might just be more important to the butterfly's survivability and propagation than the effects of thermoregulation. And now Dylan will go over our findings with undergraduate research.
What we learned was how to use software such as ImageJ, idigbio, and geolocate, mentioned previously. We also learned how to search, read, use, and cite scientific literature. In doing so, we also learned how to write posters, as well as how to write scientific literature and scientific papers. And not only does undergraduate research teach us how to learn, but it also teaches us how to question and discover things occurring in the natural world around us. Thank you for taking the time to view our presentation. We greatly appreciate it.